afternoon. My name is Michelle Ashton. I'm the Director of Communications for the Fish and Wildlife Foundation of Florida. And on behalf of our entire board and staff, I want to thank you so much for taking time out of your busy day to join us for a conversation about mussels. A couple quick housekeeping items before we get going. Could you please double check to make sure that you're muted and remain muted throughout the duration of our time together today? The exception is during the question and answer portion at the end. You can use the raise your hand feature, during which point I'll ask you to unmute and you can ask your question directly to our presenter. The second option, if you have a question, is to just drop it directly in the chat and I can ask that for you. That feature is super helpful if you have a question pop up during the presentation and you want to make sure you don't forget it. I'm very excited today to be able to introduce Olivia Maggiacomo. She's an FWC research biologist specializing in shellfish. She's going to share progress on a project growing mussels to improve the water quality in Lake Trafford, which is a prized fishing lake in South Florida. The foundation has contributed over $190,000 to this project over several years, thanks to our Conserve Wildlife plate. So if you're an owner of the Bear plate, both the redesigned or the older version, your $25 contribution has gone to this project, and we thank you so much for that. Olivia, if you want to get us going.
Sure, hi. Let me start sharing my screen. Right, so my name is Olivia Maggiacomo. I am a biologist with the Florida Fish and Wildlife Conservation Commission. I've been working on this project for the past 2 years. Before that, I worked on oyster aquaculture and Is this working?
Yes, I can see it perfectly.
Okay, sorry. It keeps popping up that someone's requesting control of the screen. Okay, so before this, I worked on oyster aquaculture and restoration in Louisiana. Okay, so today I'm going to be talking about what freshwater mussels are, why they're important. I'll talk about the life cycle and how we produce them at the hatchery, about our Lake Trafford stocking project, and the project goals and accomplishments that we've made so far. So first, what are freshwater mussels? I know it's not something that most of us are familiar with unless you're working in the field, but they are a really important part of aquatic ecosystems. So freshwater mussels are bivalves. They're mollusks that have 2 shells that open at a hinge. They're benthic animals, so they spend most of their life as adults buried in the bottom of lakes and rivers, with just the tip of their shell poking out so that they can filter feed. And for filter feeding, they filter out phytoplankton and other particles from the water using their gills. But why are they important? The biggest one, and the one that's most relevant to this project, is that they help filter the water. So as those mussels are filter feeding, they pull microscopic particles from the water and deposit them on the bottom of the lake. This results in clearer water and improved water clarity. They also help stabilize the substrate. So when mussels bury themselves in the bottoms of lakes, they help keep the soil, or substrate as it's also called, in place. This helps prevent erosion, which can worsen water clarity. They're also an important food source for other animals that live in the lake. Many species of fish, otters, raccoons, and a lot of different wading birds all rely on freshwater mussels as food source. Shellfish act as biological filters. So as they're filter feeding, they're removing particles from the water, and that can help improve water quality and water clarity. The picture here that we've got, I believe this is Chesapeake Bay, and it's an oyster example, but it's similar for freshwater mussels as well. Both of the pictures started off with the same water in both tanks, but one of the tanks has oysters in it, and after a few hours, the oysters have filtered the water so that instead of being murky, now it's clear enough that you can actually see the oysters inside. Without clear water, eelgrass and other submerged vegetation can't access enough sunlight to grow. So cleaner and clearer water filtered by those mussels means better habitat for eelgrass and for other sport fish and other animals that live in the lake. Unfortunately, freshwater mussels face quite a few threats. Florida is home to over 60 species of freshwater mussels. 16 of those are federally listed as endangered or threatened. In North America, 55% of mussel species are extinct or imperiled, which is a lot, especially considering that only 7% of the continent's bird and mammal species are extinct or imperiled. Freshwater mollusks are the most critically endangered group of organisms in North America, and that's due to a number of factors. Habitat degradation due to pollution and siltation, so fertilizer runoff, eutrophication, any pollutants that wind up in the water. And siltation is when there's, if the mussels are getting buried by excess amounts of sediment deposit or muck. Habitat fragmentation from dams. So freshwater mussels, part of their life cycle, they hitch a ride on fish. And if those fish are migratory in a river and a dam blocks the fish from migrating, the mussel isn't able to complete its life cycle, so it's lost its host fish. Habitat alteration and removal from dredging. So if you dredge a body of water, you're removing that layer of substrate from the lake. And if that substrate has mussels in it, you're removing those animals from the lake. And then competition from invasive species. Most of North America has most of the United States has problems with zebra mussels, which are outcompeting native species. We don't have them in Florida yet. I think it's too warm for them and hopefully it'll stay that way. But corbicula, or the Asian clam, is another invasive species that's in Florida. So how can we help? By stocking hardy native species of freshwater mussels, we can help restore native shellfish populations and improve water quality and clarity for other species that live in these lakes. So by raising hardy species that can tolerate poor water quality conditions, they can help clean up the water for more vulnerable species as well. Freshwater mussels have kind of a crazy life cycle. Most of us, when we think of shellfish, we think of the adult form that it's got a shell. It just kind of is mostly sedentary. It might have a foot to scoot around a little bit, but for the most part, they're not moving a lot. But freshwater mussels in particular actually have a parasitic part of their life cycle where they need a fish or other host animal in order to transform into adults. So for their life cycle, it starts when a male mussel releases sperm into the water column. The female mussel siphons it in and uses it to fertilize eggs that she holds in these pouches in her gills that are called marsupial gills. Those eggs develop into parasitic larvae called glaucidia, which we've got a picture of on the left. And those parasitic larvae need to attach to a host fish in order to transform into juvenile mussels. So once the baby mussel has spent anywhere from a few days to a few weeks on the fish, it goes through that metamorphosis, it drops off of the fish, it buries itself in the substrate, and it starts to filter feed. Those juvenile mussels continue to grow for a few months until they reach sexual maturity. And some species of mussels can live for up to 100 years, so they're very long-lived animals. But in order for those larvae, those glaucidia, to transform, they need a host. And since the mussels can't exactly chase down their host fish, they have to get creative on how to attract them. There are two common types of host attraction: lures and mucus nets. So on the left, that lure that looks like a fish or a minnow is actually a part of the mussel. And that's just above those marsupial gills that have the larvae in them. So that mussel can actually flick that lure when a fish gets close, and it acts as a bait fish. When the fish gets closer and it bites that bait fish, the mussel spits out its larvae into the fish's mouth. The larvae attach to the gills or fins of the fish, and they're able to go through metamorphosis that way. Another way that they can go through that transformation is using a mucus net. Now, it's kind of difficult to see in this picture because the mucus nets are clear, but you can see in the picture the glaucidia that are embedded in that mucus net that the mussel spits out. When a fish, or in this case, the species is a generalist, so it can even use salamanders. Whenever the host swims through that, the glaucidia clamps shut onto the fins or gills of that animal, and are able to go through transformation that way. There are three types of freshwater mussels that we work with at the hatchery. The first is on the left, that's the paper pond shell. Those are faster growing but shorter lived species, and they get their name from that really thin shell. With that species, you can squeeze on the edges of the shell and it'll even pop open. So super thin shelled, not the best for desiccation or areas that will dry out, but they're really tolerant to mucky conditions that a lot of other mussel species would have a hard time surviving in. In the middle, we've got the Florida rainbow, Glossa amygdalum. This mussel is only found in Florida, but it's one of our native species. They have these really colorful rays when they're young, but they actually grow out of those rays as they get older. And after about a year, they turn a lot of times they'll be darker, the color of that mussel on the right. So the main way that we tell them apart is the shape. The Florida rainbows have more of a thumb shape, a rounded shell. That third species is the Florida spike, which we've just started spawning with. That species actually used to be considered several different species, but they recently lumped them all together. And that can have, it's got plastic morphology. So that one mussel can have a lot of different physical attributes. But the main one that we looked for identifying them is that we call it a Nike swoosh. It's that sharp ridge on the top of its shell. And that's a feature that a lot of other mussels don't have. So in order to spawn mussels, first we have to find gravid mussels. So gravid mussels are collected near Lake Trafford. We bring them back to the hatchery for spawning. Those mussels are tagged so that we can keep track of which mussels are the parents of which juvenile mussels, and we can keep track of where those mussels were collected from. In order to find the mussels, you actually have to get into the water and dig around with your hands, and you can usually feel for them in the first couple inches of the sediment. You can dig through, find them, and check if they're gravid by looking at their gills. So when I'm talking about a mussel being gravid, I'm referring to those parasitic larvae called glaucidia that are inside those marsupial pouches in the mussel's gills. So you can see in this picture, it's a lot more obvious when it is gravid than when it's not. So when it is gravid, those pouches are full of those larvae. So they're inflated, they're a lot more obvious, and you can see the distinct chambers on the species. This is a Florida rainbow. And on the left, you can see the mussel that isn't gravid. The gills are barely noticeable. They flatten up against the back of the shell, and they're not, they don't have any color changes. They're still that pale color, and they are not inflated. Once we have gravid mussels, we bring them back to the hatchery, and we are able to extract the glaucidia for inoculation. So we flush the glaucidia out of the mother mussel's gills using a syringe filled with water, and we flush them into a beaker. We're able to check the glaucidia for viability before we use them in a spawn. And how we do that is we take a subsample of them and we examine them under a microscope, and we can tell just visually if they're still embryos or if they're viable, fully developed glaucidia. And something that's interesting about how glaucidia know when it's time to attach to a fish. So freshwater fish are saltier than the water around them is. So one of the chemical cues that glaucidia can use is salt. So to check if they're viable in the lab, we can take a small subsample and we sprinkle salt over the glaucidia. And if they snap shut, that means that they're viable and they're ready to attach to a fish. So at the hatchery, we mainly use bluegill as our host fish. They're a nice versatile fish, and they're a common host fish for a lot of species, which makes sense because we have a lot of bluegill in Florida. So first, we take our host fish that we've collected, which since we're doing this at a fish hatchery, we have access to a large number of bluegill to use. So we take our host fish and we put them in an aerated bucket so that the water is nice and frothy. We pour the viable glaucidia over the fish, and we wait 20 to 30 minutes to allow the glaucidia time to latch on to the host fish. After they've been inoculated, we move them into a tank system of racks called a ZHAB, which is pictured on the right. And the ZHAB name actually stands for zebrafish habitat. These tanks are commonly used to house zebrafish for medical research, but they also work well for our purposes, raising mussels. So transformation is kind of hard to observe because they're microscopic and they're attached to the fish. But on the left, we've got a picture of a glaucidium on a host fish that's encysted. So it's latched onto the fish scale and it is taking in nutrients from the fish to go through that metamorphosis. On the right, you can see the transformed juvenile. That is a 1-day-old juvenile that has just dropped off the fish. And you can see it's sticking out its rather pale foot, but you can see it's starting to scoot along with that foot and try and bury itself in the Petri dish. After the mussels have transformed, we collect them from the ZHAB tanks and we move them into indoor static tanks. They stay inside for the first few months of their life until they reach 1 millimeter in length, in which case they're large enough for us to move them outside. This is the most vulnerable stage of their life cycle. So they're a lot more vulnerable to water quality, temperature swings, predation from things like flatworms and fly larvae. So keeping them inside gives us more control over that. Once they reach 1 millimeter long, we're able to move them into outdoor ponds in grow-out baskets. So these baskets have a layer of sand in the bottom for the mussels to burrow in and bury themselves. They also have mesh on the sides and top to keep the mussels in and predators out. This also helps shade them from the sun and just make sure that they're covered and reduce the amount of detritus that gets into the baskets. We measure the mussels once they reach stocking size, which for us is about 20 millimeters. So once they're large enough, we tag them with, we have these really tiny tags that we attach with super glue to their shells. And those are numbered so we can keep track of which individual mussel was stocked where and where the parents were from, just to help us keep track of our production. So we tag and measure the mussels and they are ready for stocking at that stage. So the time that it takes to reach this varies based on species and weather. Warmer weather means they grow faster, cooler weather, they grow slower. But generally, it takes them about 5 or 6 months to reach stocking size. Now I'm going to talk about Lake Trafford. So Lake Trafford has struggled with eutrophication. It was overgrown with hydrilla. It had fish kills and oxygen depletions. And it also had a thick layer of muck that was settled across the bottom of the lake that was smothering it. In the 1990s, restoration work began to help restore the lake. They removed the hydrilla from the lake, dredged it to remove that thick layer of muck. There were also plantings of native vegetation and fish stockings to try and help restore that natural habitat. You can see that after, a lot of work has been done to help restore the lake, but unfortunately, Lake Trafford still struggles with water quality and clarity issues. And because the water isn't clear enough, a lot of the submerged plants that they've planted in the lake have struggled to grow because they're not getting enough light. And that's where this project comes in. The goal of this project is to raise native freshwater mussels to stock in Lake Trafford and to restore native populations and help improve water quality and water clarity. The top picture is of some biologists on a boat on Lake Trafford, checking out sites for stocking. And the bottom picture is some hatchery produced mussels that are tagged and ready to go into the lake. For our current progress, we've tested quite a few different spawning and grow up methods, and we found the methods that work well for our setup at the hatchery, which I've just gone over. We have hatchery raised paper poncho and Florida rainbow mussels that are now reaching stocking size and sexual maturity, and we are ready for stocking them in Lake Trafford in this fall. So upcoming steps will be stocking in a few months into Lake Trafford. We're also working on increasing our production to pump out more mussels, and we're looking to spawn with more species of mussels. We've started spawning with Olivia Maggiacomo, and we've had some good test trials, but we're looking to expand that and potentially expand to other species as well. This project was made possible thanks to support from the Fish and Wildlife Foundation of Florida. I know this project and many others wouldn't have been able to go forward without their support, so I'd like to say thank you to them. And if anyone has any questions, I'm here to answer them.
Great, thank you so much, Olivia. While we wait for questions to come in, maybe you want to tell us the names of those cute animals enjoying the sunlight in your last slide.
Yes, I figured some vertebrates after the invertebrate part of the presentation. On the left is my dog Goose when he was a puppy, and on the right is my cat Lucy.
Oh, love it. So folks, remember, you can use the raise your hand feature or just drop the question in the chat and I can ask it for you. I feel like I remember there was, when the project was developing, like how best to grow mussels in that kind of environment. My memory is that this might be the first time or the largest attempt at growing in this number. Do you want to talk a little bit about that, Olivia?
Sure. So I know there's not a lot of other places that are producing freshwater mussels, especially in Florida, which is different than growing them in other climates. When we started, we were just figuring out how to spawn them in general. We didn't really have a lot of experience with that, and it's something that hadn't really been done very often. So within the 2 years that we've been working here, we've been able to set up our spawning methods, and we tested a bunch of different methods. In the beginning, we were just putting the mussels in a tank with some fish and seeing if they would spawn and seeing how that would go, which worked, but it wasn't giving us very high yields. And it was more stressful for the fish, actually. So we switched to using the bucket inoculation method. So, we've tweaked our formula over these past 2 years, but we've been able to get increased survival and increased growth, and we've had a lot of success with it, and hopefully we'll continue to moving forward.
Awesome. Mitchell is asking if there's any interest in expanding the effort to other lakes and canals.
Yeah, there are other groups that have reached out to us with an interest in getting lake mussels for their lakes. So we are considering expanding. We're currently working with other groups and advising them on that.
Great. Let's see. I don't have any other, there's any other questions that have come in. Do you,
I think Hannah Phelps is raising her hand.
Oh, I see that. Thank you so much.
Hey, can you guys hear me okay?
Yeah, perfect.
Okay, good. I was just wondering, you're talking about the gravid and not gravid mussels. Is there a certain time of year that these mussels are gravid, or can you kind of collect them throughout the year?
Yeah, so it actually depends. A lot of the data that so it depends on the species. You've got long-term brooders and short-term brooders. Some of those are gravid only at certain times of the year. And for some of the species, it's only when this particular migratory fish is in the ecosystem, like in the spring, if they're going there, the mussels will be gravid at the spring to coincide with when the fish are there. For other species, they're gravid more year round, like paper pond shells. Most mussels aren't super gravid in the summer, which kind of makes sense in Florida. But a lot of the data that we have for gravidity is based on species that are in the panhandle. So a lot of what we're doing is trying to collect data on our own on, okay, in South Florida, when are these animals gravid? So it depends on the species and the location.
Cool, thank you. Yeah, that definitely makes sense considering the biological need of the host and everything like that. So thank you.
Mm-hmm. No, thank you, Hannah, for your question. We have a question from Marvin wondering what the public can do to help. Well, I'm gonna give one plug, Marvin. If you don't own, if you have a specialty license plate, or you're, I should say, if you don't have a specialty license plate and you're in the market for one, you can get the Conserve Wildlife plate that funds projects like Olivia's. But Olivia, maybe you have some other ideas on how the public can help.
Yeah, I think supporting other research attempts that are going on with freshwater mussels and just bringing awareness is super helpful. Another thing that can be harmful to mussels is, and a lot of marine or freshwater ecosystems, is eutrophication. So if you're using fertilizer or other products on your lawn or your garden, just being careful about what's running off into lakes. So not using them before it rains, and just being careful about aquatic pollution.
Great, that's really helpful. Let's see, I'm not seeing any other hand raises. So folks, feel free to use the raise your hand feature or drop it in the chat if any questions have popped up. Ooh, I saw one hand just went up. Or Hannah, maybe.
Yep.
Hannah,
No,
you have
I've got another one.
Yeah. Okay. Yeah, please go ahead.
You may have covered this already. I had to peek out for a second, but have you all put any mussels in Lake Trafford yet? And if so, how many have you released?
So we did a trial run just to test different sites that we thought would be good candidates for just figuring out where we would want to put the mussels in Trafford. We actually had a problem where we released the mussels in on bottom bags. And so Lake Trafford has the highest density alligator population in Florida outside of the Everglades. the alligators were very interested in our bags, and we had issues with the alligators attacking those bags. So we will not be using those bags for future stocking methods, but we did briefly have some mussels in the lake.
Man, that's interesting. What other methods of stocking do you guys have besides the bags? Sorry, this is like super interesting to me. I know nothing of mussels. They might
Yeah,
become my new favorite animal.
It's not something that most people think about because they just kind of are, you don't notice when they're doing their job correctly. You know, they're filtering the water, they're just hanging out on the bottom. Floating baskets are an option, though those might attract more alligator interest. The methods that we're looking in are on bottom cages or just kind of pens and releasing them so that they can bury themselves and hide and kind of not drawing attention to them. Because I think in the, I don't think alligators normally attack mussels. I think it was just they were in this bag with floats that looked really interesting to the alligators, and there's not a lot to do in that lake. So they were just bashing the bags.
Cool, thank you.
Well, thank you. Let's see, I'm not seeing any other questions. Olivia, is there, other than finding out that alligators are interested in your mussel bags, is there anything else that's been a surprising find from you from doing this project?
Oh yeah, there've been a few things. I know some of it has been like we didn't know host fish for Florida rainbows. There were no recorded host fish. So that was one that we had to find out. Luckily, bluegill and other sunfish seem to be good host fish for that species. But yeah, when I was looking up the species before we started with it, there were no confirmed hosts. So we were trying to figure that out just based on similar, in that other mussel species in that genus tend to use sunfish. So we were hoping it would carry over, and it seemed like it did. We also were surprised about just the difference that growing them outside had on their growth. They grew a lot faster once they reached that 1 millimeter and they were large enough for them to put them outside. They put on a ton of growth when we moved them outside into the floating baskets. So that was a surprise for us as well.
Super cool. Thank you so much. Well, unless we have any other questions, Hannah, any other questions on your end, since you've had some really good ones?
No. I think I'm good, but thank you.
You're good? Okay. Well, we'll give folks another couple of minutes in case something pops up, but I just wanted to, again, thank you, Olivia, and everyone else for taking time on the call. If there's, if you guys have any questions that do pop up later, feel free to drop me an email. You can reply to, if you received the email yesterday with, the information for this, you can reply directly to that. My email comes up there, or go to our contact us page on our website, wildlifeflorida.org. You can reach me there as well. And I'd be happy to pass on any questions to Olivia, get you some answers. We also have information there about on our website about the grants we've given out for this project. So if you just want to do some digging there, that information, we've also done some news stories and blog posts about this. This is one of my favorite grants, just personally. I love following the progress because I think it's just so interesting to using nature to help heal nature. I love that. So if you want to do some more reading or researching about this project, there's some resources on our website, and just really want to thank Olivia for your time and the excellent presentation, and for sharing photos of your pets with us. Was there anything, Olivia, that popped up for you during the conversation you wanted to make sure we had a chance to talk about today?
I don't think so. Just wanted to, thank you for setting this up so that we could answer some questions. And if anybody does have questions, we'd be more than happy to answer them. You can contact Michelle or Richland Fish Hatchery. And, we love talking about the project.
Great. All right. Well, I want to thank again everyone for joining us, and for Olivia. And if you have any questions that pop up after we sign off today, then don't hesitate to send them along. And we were recording this, so if you want to share it with colleagues, that would be great. I'm also going to drop in the chat here in a minute a survey. If you get a chance to take the survey and just give us feedback on what you thought worked, what you'd want us to talk about in the future, that also helps us. So if you have a minute to hang out and grab that chat, or we'll send it in the email with the recording as well. Great. Well, thank you everyone again. I really appreciate it. And we'll look forward to talking to everyone next month. Thanks, Olivia, again.
Thank you.
Bye, guys.